Hi everyone, my name is Sianji, and in this video I will be presenting an example of regulation of gene expression that occurs through intergenic transcription. The article I will be discussing was published in the journal Nature and is one of the first to identify a regulatory role of intergenic transcription. So intergenic transcription is the transcription of DNA sequences found between genes. The purpose of this type of transcription is not very well understood, but it's thought to play a variety of different roles. In this paper, the researchers demonstrate an example where intergenic transcription serves to regulate the expression of other genes, and they use a variety of different genetic constructs to determine the exact mechanism through which this occurs. Specifically, the authors studied the CR3 gene in Saccharomyces cerevisiae. So S. cerevisiae is a yeast species in which the CR3 gene encodes the enzyme phosphoglycerate dehydrogenase, which is required to catalyze a step in the biosynthesis of serine. Normally, transcription of CR3 is repressed in serine-rich media, and the authors sought to determine what types of factors are involved in repressing the transcription of CR3 in these conditions. They performed chromatin immunoprecipitation, or CHIP analysis, on the CR3 gene and its surrounding areas. And what they found was when the CR3 gene is repressed, the DNA sequence upstream of it is associated with factors involved with active transcription, such as TATA binding protein and RNA polymerase II. Transcription run-on analysis also showed that there's a high level of active transcription that occurs in this region. So to determine the purpose of transcribing this region, the authors compared the DNA sequence found here in S. cerevisiae with 4 yeast species that are closely related to it. And this sequence comparison revealed that this DNA sequence contains a second TATA element upstream from CR3's own TATA element that is perfectly conserved in all 5 species of yeast. The analysis also revealed conserved sequences which the authors proposed function as CR3 UAS, or upstream activator sequences, based on the results of deletion analyses they performed. Now, outside these two conserved elements, there's no other sequence conservation between the yeast or an open reading frame, suggesting this region doesn't encode a protein. Because this region is transcribed, and as I'll shortly show, represses transcription of CR3, the authors named this the SRG1 gene, or the CR3 regulatory gene 1. And to test the role of this gene, the authors created a mutation in the SRG1 TATA element. So in this mutant, transcription does not occur. And as you can see here, when there is no transcription of the SRG1 gene, CR3 is no longer repressed. So in other words, the transcription of SRG1 is necessary to repress expression of CR3. The researchers sought to assess the mechanism by which SRG1 represses CR3 and hypothesized that it could be acting through one of these 3 mechanisms. Firstly, the RNA product model, in which the SRG1 RNA product itself would be required for the repression of CR3, perhaps by interacting with other proteins. So an example of this we've already seen in class is the Zista RNA that acted on a X chromosome to inactivate the entire chromosome. In the second promoter competition model, the SRG1 promoter would outcompete that of CR3 for transcription factors, meaning transcription of CR3 wouldn't occur. And thirdly, in the transcription interference model, the transcription of SRG1 across this region, which contains CR3 activating sequence, would block the binding of transcription factors necessary for CR3 activation. In the next couple of slides, I'll be going over the experiments the researchers performed to test these 3 models and the techniques they used. So to test the RNA product model, the researchers made diploids in which one copy of CR3 was replaced with URA3, such that expression from one specific SRG1 promoter could be assayed. So in one diploid, URA3 was next to a functional SRG1, and in the other diploid, it is adjacent to a mutant SRG1. What they found was when the only functional SRG1 is in trans to the URA3, the URA3 transcript can be detected in that it's no longer repressed. So in other words, SRG1 cannot repress when it is in trans, so it doesn't repress through a trans-acting RNA product. Next, the authors test the promoter competition model. Normally what happens is when the SRG1 transcript is transcribed, it goes through this region where the SIR3 promoter is, and thus it's not available. So to test this model, the authors constructed a construct in which both promoters are available and functional. They replaced 150 base pairs of SRG1 with the His3 gene and its terminator sequence, such that transcription of SRG1 still occurs, but it's terminated before it reaches this UAS. And what they found was in this construct, SR3 is no longer repressed. This allows us to rule out the promoter competition model, because if SRG1 did indeed act through this mechanism, we would expect that transcription factors would still be more attracted to this promoter, and expression of SIR3 would not occur. Finally, to test the transcription interference model, the authors wanted to see if transcription of SRG1 would block the binding of transcription factors necessary for SIR3 activation. Because activators of SIR3 have not been identified yet, the researchers constructed strains in which the SIR3 UAS, shown in the blue boxes here, was replaced with binding sites for GAL4, shown in the green here. This means that in order for SIR3 to be expressed, GAL4 would need to bind these sites, and as we know from our course content, GAL4 would bind and drive the expression of the gene when cells are grown in galactose-rich media. So in a construct with a wild-type SRG1 gene, transcription occurs across this region. When cells are placed in a galactose-rich media, where we would expect GAL4 to bind its sites and activate expressions, the researchers saw that there is no SIR3 transcription detectable, thus SIR3 transcription is not galactose-inducible, indicating that GAL4 can't access its binding sites. In contrast, in an SRG1 mutant, where transcription doesn't occur, SIR3 transcription is galactose-inducible, in that the GAL4 protein can now bind this site. So thus, these results show that SRG1 transcription represses SIR3 expression by blocking the binding of activators required for SIR3 expression. Although this study does a great job of showing an example of intragenic transcription, how it can be regulatory, and how this regulation can occur through a transcription interference mechanism, there are some limitations to this study. Firstly, the study relies on the use of the GAL4 construct to characterize the interference, but a full understanding of how regulation occurs endogenously will require finding and characterizing transcription factors for the actual SIR3 promoter, so that we can test that SRG1 transcription does indeed block the binding of these activators. Further studies could also focus on how the SRG1 expression is activated and how these activators are regulated. Although the authors don't comment on it, I would assume these transcription factors could be regulated by serine, such that when serine levels are high, they bind the activator of SRG1, and in a way, these activators indirectly act as repressors of SIR3. Further studies could also focus on how this method of repression is affected by amino acid starvation, or how it interacts with other mechanisms of SIR3 regulation, such as the SWI/SNF complex. So to summarize, the findings presented in this paper were one of the first to show how intragenic transcription can serve a regulatory role. The authors clearly demonstrate a variety of genetic constructs to test different mechanisms of regulation. This has been an important theme in our course from the beginning, to not only understand the processes of regulation, but to gain a better understanding of how experiments can be designed to test hypotheses relating to gene expression regulation. With that, I'd like to conclude this video, and thank you very much for listening.